Good afternoon, everyone, and welcome. We'll get started in just a moment here, but before we do, just a few details for you. We are recording the event and we'll be posting this video up on the Basic Science website in about a week. So if you'd like to hear the talks again, or if you know someone else who might be interested, you can view, you'll be able to view this video and other videos in this season and past seasons at basicscience.berkeley.edu. We'll post that URL in the chat for you as well. We are going to keep the audience muted throughout the event, but we do want to hear from you. So if you have questions for any of our speakers, please post them in the chat. We'll be doing Q&A throughout the event, and we'll try to get to as many questions as possible. And with that, it's my pleasure to introduce the Dean of Biological Sciences, Mike Botchin.
Good afternoon, everybody, and welcome everyone to Basic Science Lights the Way. I fit into a number of categories as a molecular and cell biologist, a biochemist, a virologist. So I'm particularly delighted to be here this evening to introduce you to a number of faculty from our Integrative Biology department. And I'd like to add that actually here is at Sea Ranch. So due to the marvels of the internet, I can be up north and still with you. We launched this series of virtual talks in the midst of the pandemic to stay in touch and to celebrate the breadth and depth of basic scientific discovery at Berkeley. We've decided that we really like this format since it allows us to reach new audiences and to showcase our amazing faculty and students. I've even started to recognize repeat attendees. This series is intended to shine a light on basic science. By basic, we don't mean simple, but rather fundamental and foundational. This is the type of curiosity-driven research that allows us to devise or revise theories and purport general truths. While it's important to apply our science to solving problems and finding therapies or cures, without basic research, there can be no breakthroughs or innovations that lead to applications and accolades. Once we get underway, we will encourage you to post your questions for any of our speakers in the chat box. We'll answer as many of them as we can. While we can't possibly be comprehensive in 1 hour, we hope to inspire your curiosity to learn more about your leisure, at your leisure. Tonight's topic is creatures of California. As billed, the Golden State is a hotspot of biodiversity, and Berkeley's biologists are investigating its remarkable flora and fauna. Our moderator this evening is Carolyn Williams, an associate professor of integrative biology. Carolyn has given a wonderful talk previously in the Basic Science series, so she's a repeat performer here. Carolyn's research interests span evolution, physiology, ecophysiology, metabolism, insects, winter adaptation, and thermal biology. The Williams Lab studies the evolution of metabolic physiology in ectotherms, using insects as models with implications in understanding how to predict organismal responses to global climate change. Okay, over to you, Carolyn. Looking forward to the evening. Bye.
Thank you, Mike. It's such a pleasure for me to participate in today's event. So as Mike said, I had the pleasure of presenting at one of these events myself, and I'm really excited today to introduce you to some of my colleagues and their work. So when I tell people that I'm in the Department of Integrative Biology, I often get kind of curious stares and the question, what's integrative biology? So I want to tell you, and then my colleagues will show you what integrative biology means to us. So integrative biology means using the full power of biology and interdisciplinary science to understand our origins, improve our quality of life, and conserve our natural ecosystems. And from the level of the genome and the tiniest cells to the largest organisms on the planet and the entire ecosystems that sustain us. Researchers in IB, as we call our department, work in the field, in our world-class museums, and in pioneering labs to drive breakthroughs in the natural science. From the bottom of the ocean to the crowns of the tallest trees, we study organisms across the entire tree of life to achieve an integrative understanding of what makes us who we are in this web of life that sustains us. So today we're going to hear from 3 inspiring IB faculty members, Todd Dawson, Rory Bowie, and José Pablo Vásquez-Medina. They will illustrate the power of integrative biology to teach us about some of the most iconic creatures of California, redwoods, California condors, and elephant seals. So I hope that you'll enjoy these amazing stories of California creatures. At the end of each talk, we'll address questions from the audience, so please add your questions in the chat. So our first speaker today is Professor Todd Dawson. So Todd is a professor of integrative biology. His research focuses on the relationship between plants and the environment using techniques from physiological and evolutionary plant ecology, remote sensing, and stable isotope biogeochemistry. Tonight, Todd will be talking about California's iconic coastal redwoods. Over to you, Todd.
Great. Thank you, Carolyn, for that introduction. Thanks, Mike, for hosting this event. And thanks to all of you for joining this event this evening. I'm really looking forward to telling you a little bit about Redwoods, the greatest show on earth. It's a provocative title. It's getting you to think a little bit about the iconic species of redwoods that we have in California. And in the short time that I have today, I'm going to share a few of the highlights that we have been discovering in redwoods in the 30+ years we've actually been researching them here in California. Many of you may know that we actually have 2 redwood species here in California. Our coast redwood, which is shown in the blue distribution map here along the coastal strand from southern Big Sur all the way up into the Oregon-California border. And then the giant sequoia, which is located in the south central Sierra Nevada in about 70 separate groves. And then it extends north in a couple of groves that make their way close to Yosemite. In the work that we've been doing on redwoods, we've uncovered some really incredible facts. The first of these, of course, is that they are the tallest and most massive trees on Earth. The tallest tree is a coast redwood, 380 feet tall, and the largest redwood is the giant sequoia, which is 4.2 million pounds. These trees are also extremely long-lived, living somewhere between 2,000 and 3,000 years plus. And mature leaves can have up to 3 billion leaves. And to give you a perspective, that would be distributing all the leaves on a single tree over a football field to about 4 inches deep. A single tree can also store an amazing amount of carbon. And in fact, somebody did a calculation and said the General Sherman tree, one giant sequoia, could, if you took the lumber from it, you could build a box that would contain the Queen Mary ship with a lid. And one other thing is that redwoods have occupied the earth for a very, very long time. So they've been exposed to a wide range of environmental and climatic variability. Given some of these facts, we have to ask some really fundamental questions. How do these trees grow so large? How is it that they live for so long? And how is it that survived or even thrived under past climatic changes? And what will the future change? In other words, if we can answer these questions, can we also ask the question, are redwoods actually breaking fundamental rules? Before we can actually even address these questions though, I want to raise your attention that there is a real challenge here. And that is, how do we study an organism that's 70,000 times larger than you and I? The drawing on the right shows people at the base for scale. Well, what we've had to do is innovate and really develop whole new ways of doing research on an organism this large. We have very skilled teams of people that are not only climbers and not afraid of heights, but they also have to be engineers as well as biologists. It's challenges to be able to develop new tools, new sensors, and new ways of using those sensors to sense the way these organisms function in their natural environments. And then we must work all the way from the leaf all the way up to the entire landscape and be able to integrate our information on basic biological processes even into the entire forest. Well, I can't take you into the redwoods tonight, but I can show you a few pictures to just emphasize how massive these organisms are and what a research challenge they actually are. In this picture here, we're looking up into a coast redwood that's about 1,500 years old. You see the climbing rope off to the right. And one of the things that you see is there is an entire ecosystem in the crown of this tree here. Soils develop, entire trees and other plants are rooted in those soils. And we even have endemic spiders and amphibians that spend their whole life in these ecosystems that are aloft in these trees. When we begin our research, we start out with basic metrics. We have to get out there with just simple tape measures and be able to quantify the allometrics, we call it, of a tree, to be able to quantify how much carbon is there, how many leaves are there, et cetera, et cetera. And this forms the foundation for some of the research questions that then we can go on to ask. We need to ascend these trees and install sensors from all the way from the base of the tree, all along the tree itself that monitor microclimate, monitor physiology. And then we even have to ascend the trees to the top where we can actually sample foliage and wood and cones to be able to quantify chemistry, a number of other physiological aspects of the trees themselves. One highlight that has come forward in the work that we've done is the incredible relationship that coast redwoods have with fog. As you know, fog is a summertime phenomena in the Bay Area. It is a water resource that is in there when rainfall is not present. And one of their important discoveries we've made is that when fog accumulates on redwood foliage, some of it drips, but a lot of it stays behind. And when it stays behind, if we look at the microscopic features of the leaf, we see that the leaf surfaces are colonized with fungi. Those fungi penetrate the leaf stomata, which is the small pore that allow gases to exchange between the atmosphere and the inside of the leaf. And those mycorrhizal fungi, those fungal hyphae shown here as those white strands, are actually acting as a water wick that allow water from the outside of the leaf to make it on to the inside of the leaf. When that happens, that changes the entire physiology of a coast redwood tree. Shown here on the vertical axis is water moving through a tree along over several days. If you look at the diagram over on the right-hand side, I've got some arrows in the animation showing you in the daytime, water's flowing at the top, the bottom of the tree out into the atmosphere as we normally expect. But if you look at the green arrow, when fog comes sweeping in at night, one of the things we begin to see, and you can watch the animations over on the right-hand side, is we get reverses of flow. Water's coming into the leaf, into the trunk, and that continues as it goes all the way down to the very base of the tree, refilling this remarkable reservoir. Now, these microbes that are kind of responsible for changing that physiological behavior are not just in the crowns, but they're also in the ground. And one of our most recent discoveries is on the roots of redwood trees, we found a very new organ that we are now calling the Rhizonode. And in that Rhizonode is a remarkable microbiome. It harbors a wide diversity of fungi, as shown in the graph in the upper right. Inside the Rhizonode, there's 20 times more fungi than there are on the root itself or out into the soil. It also harbors bacteria. And the coolest thing that we've been able to find is that when we do the DNA sequencing of that bacteria, they revealed that the species of bacteria are known to actually fix nitrogen and confer drought tolerance to the trees themselves. A remarkable symbiosis that was just discovered 2 years ago. We also climb into these trees and use the long tree ring records to reveal things about how the trees have responded to climate in the past. We can look at the ring widths and we can look at the chemistry of those rings, particularly the oxygen isotope composition, which will tell us something about how climate and physiology has changed over vast periods of time. When we do that, we can begin to map this out. We can be able to reconstruct periods of wetter or drier, such as through the Little Ice Age shown at the top of the slide here. And one of the things that's revealed is that in the past, people have said the Little Ice Age was a very wet period throughout. Our records are showing that, in fact, it was wet for a while, and then it was highly variable after that in the coastal climate of California. If we also look at a period from 1670 to 1870, it was very wet, but then it became very cool, and is something that was never revealed in coastal climate before. And finally, of course, the well-known Anthropocene warming is also revealed in the tree ring records of redwoods, which has been shown in many other records worldwide. We've taken information that we've been able to collect from the trees, and now we've flown over these forests, and using hyperspectral sensors, we can actually now connect our physiological information to remote sensing information. And I can show you where trees, which are red here, are stressed, and where trees that are not stressed, which are the blue and yellow trees themselves. So this information has allowed us to scale up to the entire forest. So redwoods are, in fact, breaking the rules. They've really taught us some remarkable things. They've taught us that water uptake doesn't conform to anything in your botany textbook. And that water that comes in is actually influencing the amount of water every single tree uses every single day. Tree ring analysis have allowed us to reveal undiscovered aspects of California's climate and climate change. And most importantly, we see that redwoods are harboring now a new microbiome of fungi and bacteria that are really changing the game. And they may even become the peas of the forest. So with that, I'll end. I'll thank you, and I'm happy to take questions.
Wow, thank you, Todd. That is just amazing. Those are amazing organisms, and you have had to develop equally amazing tools to study them. So I'm looking forward to hearing questions from our audience. So feel free to post those in the chat. But while I have a minute, maybe I can ask one of my questions myself, Todd, of what is one of the most important new tools that you had to develop to study these amazingly large organisms?
I think the most probably influential tool that we've had to develop, Caroline, is bringing in the chemistry of isotopes into being able to tell us about how leaves function and how, when the climate changes, how that change in leaf function gets recorded in the wood, in the tree rings within a redwood tree itself. So it really was coupling chemistry with physiology and also climate science.
Yeah, it's amazing the way that the trees can then provide that window into the past that can enable us to see past climate states that I guess would be pretty hard to reconstruct without that kind of record.
Absolutely, I agree.
Yeah, great. Okay, so we've got some audience questions coming in here about the status of the sequoias in King Canyon that burned. And I know you've had some researchers focused on fire adaptations of redwoods. So
exactly right. And this summer was, of course, a very challenging summer for the giant sequoia. It burnt through one of the most amazing groves in Sequoia Kings National Park. Fortunately, the fire service did an incredibly good job of protecting at least some of the oldest trees, the most magnificent trees in Giant Forest Grove, but we lost a lot of very old trees as well. With climate change, we're seeing things are getting hotter, they're getting drier, and it's all happening faster. And this is having quite negative effects on both the coast redwood and the giant sequoia.
Yeah. It's a sobering thought, isn't it? But luckily we've got people like you working on the problem. So there's a question from Chris Cook here, that how does a tree that propagates by root sprouts So not by not having the possibility for genetic change, adapt to these climatic changes?
So it's a good question. There is a bunch of what Chris is saying is asexual reproduction, so sprouting from the base of the tree or sprouting from the branches. But there is some sexual reproduction as well. So there is cross-pollination among different trees. And so there is some genetic variation that we actually do see in these trees. So there are also other mutations, which we call somatic mutations, that come up in these asexual tissues of redwoods. And some of those somatic mutations actually confer some really interesting adaptations for the trees themselves. Without going into further detail, that's sort of what lies underneath that question, or an answer to that question.
Great. Do the microbes that you've discovered interact with the mycorrhizae?
They absolutely do. Great questions. We see that the microbes in the soil, around the outside of the root, and then in that rhizonode are all interacting. And in fact, some of the microbial biologists that I've spoken to, there is a term that they use called quorum interactions, basically where the fungi and the bacteria are working almost together and influencing each other in positive ways by exchanging resources and also exchanging resources with the plant that is actually hosting them. So there's some incredible microbial and microbiome research now that has to be done and followed up from this initial discovery that we made just a couple of years ago.
Yeah, absolutely. So, and last question I think that we have time for is, do you have any idea what fraction of the tree energy goes into the cones or into the sexual reproduction?
It's a really good question, and the fraction really is very small. Actually, most of the carbon is going into growth, radial or height growth. Most is going into replacing leaves, and maybe somewhere between 2 and 3% of that will go into actually sexual reproduction or making cones each year. But remember, these trees make millions of cones.
Yeah, over a very long period of time.
Indeed.
Right. Okay, well, I think we might have to move on to the next speaker. There's lots more great questions just coming in. We'll come back to them at the end if we have time, but thank you so much, Todd. So our next speaker is Rory Bowie. So Rory is a professor of integrative biology and also a curator in the Museum of Vertebrate Zoology. His research focuses on ornithology and in evolutionary biology, and he's fascinated by why animals are distributed unevenly around the globe. He's known for his work on tropical rainforest birds, penguins, and the birds of Western North America. And he's also been the recipient of the Campus Distinguished Teaching Award. Tonight, Rory will be talking about California's iconic condor. Rory, can you tell us about your research?
Yes, thank you very much, Caroline, for the introduction, and I'm delighted to be here tonight. So while California is famous for its spirit of ingenuity, it should also be famous for its spectacular bird diversity. Over 600 species have been recorded in the state, representing nearly 2/3 of all North American bird species. Unfortunately, over 50 of these species are threatened or endangered. Among these is the majestic Californian condor, the largest living land bird in North America. In 1987, the last 26 individuals were brought into captivity to establish a captive breeding program, rendering the species extinct in the wild. This program continues to be successful, and by 2020, captive breeding and release of animals into the wild has built the total population to over 500 individuals. Today, I want to highlight how the integration of field biology and molecular biology can lead to discoveries that are both surprising and marvelous. Discoveries that address real conservation needs while advancing our knowledge of the unknown. Such work is undertaken by several labs in IB, including my own, where I integrate field biology with cutting-edge genomics and morphological analyses in birds. Much of my own work is indirectly underpinned by one of the great achievements of the past 20 years, the human genome. The advances in chemistry and engineering that have enabled high-throughput sequencing of human genomes have also made it possible to generate a high-quality genome for any organism of interest. In July this year, we published a highly contiguous genome of the Californian condor. This landmark paper was led by postdoc Jacqueline Robinson. Our genome assembly of the condor recovered whole chromosomes and indicates that the condor genome is less than half the size of the human genome, about 1.2 gigabases versus 3 gigabases. It's remarkable that in the last 20 years, we've gone from spending $3 billion on the first complete draft of the human genome to less than 10,000 on the condor genome. To place that cost reduction in perspective, $3 billion would enable us to sequence the genome of every living bird and mammal species on Earth today. Now that we have the condor genome, what can we do with it? One of the first things we did was to investigate the extent of genetic diversity in the Californian condor, with higher diversity indicated by these large peaks along the y-axis, and how diversity is distributed across the genome indicated here on the x-axis. While we expected to see many signs of inbreeding, given that the California condor population was so severely reduced, we instead recovered a completely unexpected result. That is that the Californian condor has a genome-wide genetic diversity as great as the abundant turkey vulture, and almost double that of the Andean condor. This unexpected outcome is a consequence of the very long lifespan of condors and the success of the captive breeding program in maintaining existing levels of genetic diversity in the species. Although condors have a high genetic diversity, they did not completely escape the impact of inbreeding. They are subject to a rare form of lethal dwarfism called condodystrophy through the expression of 2 recessive alleles. With blood samples collected by the San Diego Zoo of the 26 founders from all living condors from which all living condors are descended, we are now in the process of sequencing the genomes of all of these individuals with the hope of being able to identify the gene in which this mutation resides. We can also use this information on genetic diversity in genomes to understand changes in population size over time. In this figure, time is on the x-axis going from the present to 1 million years in the past, and on the y-axis is population size in tens of thousands of individuals. This result represents one of my chief intellectual motivations for undertaking this research on condors, because I wanted to know if condors nearly went extinct as a consequence of the last ice age, driving most of the large mammal species, including, for example, mammoths and ground sloths, to extinction. Our results reveal an 80-fold decrease in California condor population numbers over the last million years, and a smaller reduction in the other 2 species indicated in green and blue over here. These results suggest that historically, the Californian condor would have numbered in the tens of thousands, and which is consistent with their range having been contiguous across the entire United States, with their decline starting about 50,000 years ago, which preceded the peak of the last glacial ice age. So it suggests that California declines preceded the extinction of large North American mammals, such as mastodons, mammoths, and ground sloths. So what could be another explanation for the decline of condors? Well, we've started looking at fossil bird assemblages in California. And so we've turned to the analysis of fossils housed in the Museum of Paleontology on campus and the La Brea Tar Pits Museum, research that is being led by my graduate student, Mackenzie Kirshner-Smith, pictured here. Her results have revealed that vultures even larger than the California were present in California, including this magnificent beast, Teratornis maryi, whose skull is almost twice the size of the Californian condor. To place this in perspective, here is a comparison of the condor versus one of these very large vulture species. The other thing you can notice, if you look at the side-on view of these skulls, they reveal a very large nasal cavity, and it has long been speculated that this plays a role in enabling these vultures' acute sense of smell, which is how they locate carcasses on the landscape. We recently CT scanned a turkey vulture skull, and I want you to notice these magnificent scroll-like bones that are present in the nasal cavity. And this is a remarkable morphological innovation that greatly increases the surface area over which odorant molecules can bind to receptors. We also see extensive neurological innovation in the turkey vulture brain, revealed by the considerable expansion of the olfactory bulb over here, which is much larger than what we would, for example, see in the size of a chicken. We've been able to expand upon this work by CT scanning the skulls of other vultures, even fossil species, to 3D print endocasts of the brain, an example which is highlighted here for the Californian condor with the olfactory bulb indicated in green. Returning to the genomic data also reveals innovation for olfaction, with expansion of the number of odorant binding genes, in particular the V1R and V2R vomeronasal genes. Thus, we see considerable morphological, neurological, and genomic innovation in olfaction by these vultures. Finally, I leave you with the unexpected recent discovery reported widely in the news. These condors are able to reproduce without mating through a process known as parthenogenesis, which also is known to occur in reptiles and sharks. Thank you for your attention, and I would be delighted to answer any questions.
Wow, what an awesome conservation success story. Thank you, Rory. They're very impressive and amazing to think that there were even larger vultures around one day. So we've got a question from the audience here. What do you think accounts for the decline in the total condor population in the last year?
So it's a complicated question. We think the single largest threat facing condors today is lead poisoning. And of course, lead was banned in California for many years, and then within the last 2 years, that ban was rescinded. So when condors are scavenging on the landscape, they're finding the animals that hunters shot, that ran off and died. They're consuming that, and as they're doing that, they're gaining lead, which it ends up lining their stomach, and they don't pass it through their gut. And so it's possible that lead is being reintroduced into the environment, and it's once more accumulating in these condor populations. That's one speculation. It could also be related to harsh elements of some of these really dramatic changes in climate that we're seeing, these sort of extreme events.
So I noticed all the pictures of vultures you presented have bald heads. Why is it that vultures have bald heads?
It really is quite interesting, and it's most likely got to do with them keeping their feathers clean. So as you can imagine, if you're sticking your head into a somewhat icky carcass, it's important to be able to clean yourselves. What I didn't talk about today, and what we've also been able to see in the genome data, is vultures have greatly expanded the number of genes involved in the immune system. So not only do they have reduction in feathers on their head as an indirectly immune response, but they also have really innovative genomes that help protect them against bacteria invading their systems.
Wow, that's fascinating. And yet you told us that the genomes of the vultures are half the size of the human genome. Is that typical for birds? And if so, why do birds have reduced genome sizes?
It is typical for birds, and it's even more remarkable when you think that birds and mammals have roughly the same number of genes that code for proteins. And we think the reduction has to do with, as you can imagine, the larger the size of your genome, the larger the cell you have to house that genome. Of course, birds fly, so they have to be economic in everything that they do. And so somehow they found a way to cut out much of the junk DNA that we see in mammals that enables them to reduce their cell size.
Thrifty. So we have another audience question here. Any explanation for the relatively small amount of genetic diversity in the Andean condors?
Yeah, great question. And it was such a shock for us to see California condors having so much. We were so shocked that we sequenced a second genome. So the reason for we think for the Andean condor being so reduced is that it really occurs in the high Andes, which is a pretty narrow strip along South America. And so its long-term range that it's occupied is likely to have been much less than what the California condor historically occupied. And so we just think that for a much longer period of time, it's had a small population size versus the Californian condor, where the population size has crashed relatively recently.
So if you had to guess, what's the outlook for our iconic Californian condor? Are you feeling optimistic?
Well, I admire the US Fish and Wildlife Service and the San Diego Zoo for how much effort has been put into breeding these birds and how successful they've been, when you think that at one stage in 1982, there were only 22 individuals. So I remain optimistic, but I am extremely concerned that later is being reintroduced into the wild, with plenty of data having shown that it's the primary cause for modern condor declines, including being able to look at some of our museum specimens to show that they had far less lead in their feathers than 100 years ago than what we see today.
That really shows the value of those precious museum collections, doesn't it? And a related question from the audience is, can the condor survive without human help? Are they going to be able to establish breeding populations?
It's a great question. And aside from the breeding element, condors, we think, are only able to survive with food supplementation. In other words, we have to provide cows, dead cows, on the landscape for them to feed on, because there simply are not enough large carcasses of animals to be able to maintain a large enough California condor population to persist.
Right. Okay, well, I think that that's all our questions for now. So thank you so much, Rory. That was really, really fascinating. So our final speaker for the evening is Professor José Pablo Vásquez-Medina. José is an assistant professor of integrative biology. His interests reside in the area of marine mammals and preclinical models of human disease. He studies mechanisms of adaptation to both natural and anthropogenic stresses at the cellular and organismal level. So José, can you tell us about your research, please?
Thank you, Caroline, for that very nice introduction. And good afternoon, everyone. It is a real pleasure for me to be here today to talk to you about the work that we are doing in my lab to try to understand the extreme physiology of the northern elephant seal. You might be familiar with these fascinating creatures who spend part of their time on the Californian coast during their breeding and molting seasons. And the rest of the year, they spend it diving in the Pacific Ocean, where they can travel up to 13,000 miles per year. Elephant seals are constantly exposed to extreme conditions. When these animals dive, they can hold their breath for up to 2 hours. And when they are on land, they undergo these absolute food and water deprivation periods that can last up to 3 months. And you may think about other animals that undergo prolonged periods of food deprivation, such as hibernators, but elephant seals are quite unique because this food deprivation happens at the same time as other quite extreme life history events. For example, catastrophic molting. Catastrophic molting is when the animals replace the outer layer of their skin and all their fur in about 2 weeks. Food deprivation also happens during fighting, and this fighting is an essential component of their breeding strategy. Elephant seals are territorial animals. One bull can control access to up to 80 females. And this food deprivation also happens when elephant seal cows nurse their pups. This is something that lasts about a month. And again, this happens while elephant seal cows don't have access to food. So we and others have been studying the organismal level adaptations to these conditions for quite some time. And something that we have learned is that all these extreme life history events induce neuroendocrine stress responses. This is just a quick picture about the stress axis. This is a physiological response that is quite conserved among all vertebrates, and that at the end, it results on the release of stress hormones into the circulation. So we can manipulate the stress axis to try to study the effects of acute and sustained adaptation to stressors in elephant seals. And we do this by using exogenous hormones and receptor blockers. When we look at gene expression after an acute stress elevation in elephant seal, it was sort of surprising to find increased expression of genes that in humans are associated with muscle atrophy. And this was quite surprising because we know elephant seals don't experience muscle atrophy. Yes, they are exposed to these extreme conditions, but they are adapted to them. So we want to know what are the mechanisms that allow elephant seals to cope with sustained elevations in stress hormones. And of course, we are sort of limited in the number of studies that we can conduct in a large wild marine mammal. So what we did to answer these questions was we went out to the field and collected muscle biopsies from elephant seals. And these are very small pieces of muscle tissue that we can bring to the lab. And from those small pieces of tissue, we can isolate muscle progenitor cells. We can make these cells differentiate into muscle fibers. And I think this is a good model because it retains essential characteristics of the muscle tissue. These muscle fibers contract, they express muscle genes, and they have a distinctive metabolic phenotype. More importantly, these muscle fibers are amenable to genetic and pharmacological manipulation. So we can expose these muscle fibers to stress hormones and start learning about the mechanistic drivers of this amazing adaptation that elephant seals exhibit. And what we have found so far in our studies is that sustained exposure to stress hormones does induce muscle remodeling, but we also find some genes that are associated with energy metabolism. And with this model, we can actually look at the metabolism of these cells and see what pathways they are using to produce energy. So under basal conditions, they use both respiration and glycolysis. These are the two main pathways that all cells use to make energy. And this is supported by an extensive mitochondrial network and mitochondria-ER interactions that promote a high energy consumption state. Upon exposure to stress hormones, however, these cells shut down mitochondrial respiration and rely on glycolysis. And when we look at the subcellular level, we see that this is supported by changes in the mitochondrial network and the mitochondria-ER interaction that promote a low energy consumption state. So what does this mean? What this means is that elephant seal muscle cells have the capacity to switch to a low energy consumption state that promotes survival during extreme conditions. So I'm gonna switch gears a little bit and tell you about the other amazing adaptation that elephant seals exhibit, and that is that they can hold their breath for up to 2 hours when they dive. And actually, when they're on land and they sleep, they can also hold their breath for up to 30 minutes. And they do that by using two main strategies. One of them is to have more oxygen on board, and more oxygen means having more blood, having more red cells in their blood, and having more oxygen in their tissues. They can also use oxygen more efficiently. When they are in diving mode, they decrease their heart rate, and they also decrease the amount of blood that goes out of the heart. At the same time, they direct blood flow to the brain mostly and a little bit to the heart. So this means that a lot of peripheral tissues are completely cut off from the circulation, which to me is quite amazing, right? Because this actually shows that these tissues are highly tolerant to reduce blood flow. And this is a condition that we know in humans is super detrimental. think about a heart attack or a stroke when oxygen is not being delivered to tissues, and this is life-threatening for us. But for elephant seals, this is not a problem. This is actual data that I'm showing you here of an elephant seal diving off the California coast, performing routine 30-minute dives. The black tracer indicates oxygen levels in the arterial circulation of these animals. And you can see that these elephant seals use up all the oxygen that they have on board. So their blood vessels are quite hypoxic. They have very, very low oxygen when they're diving. So how can we study this? We want to study this. We want to know how they do it, right? So, of course, we have very limited access to vascular tissues from elephant seals. So what we've been doing in my lab is we go to the beach and we collect expelled placentas. And placentas are a vascular tissue that elephant seals don't need anymore. So from these placentas, we can dissect arteries, and from these arteries, we can isolate endothelial cells. These endothelial cells are the cells that line the inside of the blood vessel. So these are the cells that are faced with these changes in blood flow and oxygen tension. We can grow these cells in the lab under flow, and we can expose them to low oxygen. And what we have learned so far, we're comparing the responses to similar human cells, is that these cells are highly tolerant to hypoxia. They are also highly tolerant to oxygen stress. When we actually expose them to hypoxia, they decrease expression of inflammatory genes and increase expression of antioxidant genes. So, of course, this is cool, but the question that remains is, can these strategies have translational value for the treatment of potentially environmental conditions in humans? Well, I'm gonna leave it there. Thank you very much. Happy to answer any questions.
Thank you, Jose. Another truly fascinating creature of California. So please go ahead, put your questions in the chat box. So those cell culture models that you have developed are really an amazingly innovative method to study an intractable problem. I wondered if you were planning to use these in other systems for other large diving animals or any other contexts.
Yes, thank you. That's a great question, Caroline. We actually have some support to do this in an Antarctic species that also dives very deep and for very long periods of time. That's the Weddell seal. And the advantage of that is that we can also conduct in vivo studies, right? So we can have both and look at the organismal response and the cellular response and see how translatable this is.
Yeah, the limitation advantages of in vivo compared to these cell culture experiments.
Yes, I couldn't get that. I'm so sorry. It sort of
no
like
problem. You mentioned conducting similar experiments in vivo. What are the limitations and advantages of doing it in vivo versus in cell culture?
Yeah, well, I think they are both powerful approaches that can help answer different questions, right? I can conduct genetic manipulations in these in vitro models, cellular models, but I currently cannot do that in a live marine mammal.
I imagine it's difficult to get the elephant seals into the lab as well.
Yes, I mean, we have done it in the past through collaborations with colleagues at UC Santa Cruz, but yeah, it is quite difficult to deal with these large animals, right?
Yes, I can imagine. The little beetles that I work on are much easier. So that's really great. We've got some audience questions here. So that's an amazing ability to hold their breath during sleep. Does this raise some potential treatments for sleep apnea in humans?
Yes, I mean, what we have seen is that they have this mini diving response that when they sleep. One of the differences is that in humans, sleep apnea is obstructive, right? And in elephant seals, it's a response that's controlled by the brain. So that's something that we want to think about a little bit more. But yeah, I mean, at the cellular level, the cells are experiencing low oxygen, right?
Yeah. It's another audience question. Can elephant seals drink seawater? If not, how do they go months without access to fresh water, especially nursing females?
Yeah, that's a great question. So they don't they drink water when they eat, because they just open their mouth and they feed. When they are on the beach, they don't drink seawater. We don't believe that's something that they do. They do produce metabolic water, and I think that's enough. But that's another amazing adaptation, right? How can they change their metabolism and be able to survive these periods that are very energetically demanding?
Yeah, absolutely. So another audience question, elephant seals also went through a population bottleneck in the past. Is it known what their genetic diversity is like?
Yes, that is a great question. Elephant seals were almost extinct. There were less than 20 animals left in Guadalupe Island in Mexico, and now the population has recovered to over 150,000 animals here in California in the North Pacific. But the genetic diversity is very low.
Right. Yeah. Well, hopefully they'll bounce back in the future. Do you think they've got a good shot?
Yeah, I mean, they do all these amazing things that I think, some sort of like prepare them to cope with extreme conditions.
Yeah. Great. Well, thank you so much, Jose. So that's the end of our conversations with speakers. We do have a little bit of time for questions that can be directed to any speaker or to the group as a whole. So feel free to type any general questions in the chat. So I wanted to ask a question of all three of you. Where does Berkeley research and your own research in particular fit into the solutions that we need to solve the problems that society is facing today? Feel free, anybody, to start.
I think one of the wonderful things about Berkeley is that because of its size, it encompasses people from very many different disciplines. And one of the great things about integrative biology, because of the breadth of the department, is we have faculty who touch on many different disciplines. And we know that we cannot just keep doing the things the way we have done them in the past. We have to look for new ways to innovate and new ways to bridge things. And so I think this ability to interact with new disciplines, think of new ideas through bio-inspiration, thinking about some of Todd's work in trees, or how to bring back charismatic animals that the public can identify with, are all wonderful examples of how we can think about innovating for the future.
Thank you, Rory.
And I would add, I think some of the basic discoveries that we're making here in integrated biology have direct applicability to some of the big global problems that we face today, like biodiversity loss, climatic change, environmental impacts. And everybody's research that I see across the campus more and more has applicability to helping basically provide information that will eventually lead to solutions and potentially help us solve some of these global problems that we're actually seeing across the world.
I agree with you both. I see that shift myself towards more emphasis on research for a purpose and realizing we need to close that gap between basic research and translational and applied research. And I think all three of your talks were really, really wonderful showcases of that. Right, so we had a few questions that we didn't have time to get to. Oh, here, maybe we'll have one more general question. So there's an audience question. We're losing species at a great rate with climate change as a major problem. Is there hope for the future? This is something I hear a lot, and it's a very difficult one. So does anyone want to take that question?
Well, I'll tackle it. I think there is hope for the future. I mean, I remain optimistic that our science is making a difference in what I just said a moment ago in helping us identify solutions. But it's also true that humans have to change their behavior. If we're going to keep our biodiversity rich, our ecosystems vibrant, that means that we can't keep doing things the way we've always done them. And it's a hard thing to face, but I think humanity has to realize that, you know, more of the same is not working. And if we're going to find solution and protect species, we have to be able to make some sacrifices. So, you know, we're part of the solution, but we're also part of the problem.
Absolutely. There are many possible paths through a difficult future, and the sort of research that is happening on campus can help us to pick the best of possible paths, but it definitely requires some sustained action from everybody. So an audience member asked if there are any research projects on organisms that live right on campus.
Yes, I can think about IB research on squirrels that live on campus and hummingbirds.
Yes, that's true. Our former department chair, Robert Dudley, has a little hummingbird box right in his wall of his office where the hummingbirds come to visit him.
And I believe there's some research also going on with the peregrine falcons that are living in the campanile as well. So they're one of our poster children.
There's a webcam for you, for everybody to go and watch their everyday activities. They're quite marvelous.
It's really marvelous.
Oh,
there's been quite a drama unfolding lately around the falcons, hasn't there, with the Grinnell being wounded and taken to a wildlife center. I don't know if you know the update on Grinnell.
He's doing okay. He got beat up by another male who invaded his territory. So, that was my grandma's action on campus.
Oh, Grinnell is back on campus, Jack says. Okay, that's good news. So we had a couple of questions for Todd that we didn't get to. So during an extreme drought, how is stress on the tree reflected in the state of the fungi?
Excellent question. Often what happens, and we've only seen this now one time in some of the observations that we made in last year's pretty extreme drought, some of the rhizonodes will actually fall off of the roots themselves. So the plant will actually get rid of them. That means they're getting rid of some of the fungi. And so it definitely affects the overall interaction between the fungi and the plant itself. But it also seems that it also takes the plant and puts it into a quiescent state. It doesn't metabolize as much. And so it almost seems like some sort of a defense response. But boy, there's a lot more work that needs to be done to really understand that fungal plant interaction under those kinds of stressors, such as water limitation.
Yeah, that sounds like a really important area of research. More time, Todd. We've got a question about the during the tertiary, redwoods were distributed far to the east, at least to Utah. An audience member thinks the Sierras were not there. What were they doing out there?
Yeah, I think I saw that message in the chat from our former colleague, Jer Lips. Thanks for that question, Jer. And I think what was going on during that time is, of course, to the east, the climate was a much different kind of climate. It was wetter. It was also warmer in some of those locations. And in the area, in some of those areas, then the redwoods are actually thriving quite well, even though they didn't have the fog or they didn't have the mountain climate. It was almost a subtropical climate as far as the interpretation of the fossil record goes. So a very different set of living conditions for redwoods back in the tertiary than we have today.
All right, well, thank you so much to all our speakers. To close, I'm going to hand the floor back to Mike Botchin, Dean of the Division of Biological Sciences. Mike, the floor is yours.
Well, thank you, Carolyn, and thank you, Todd, Rory, and Jose for sharing a glimpse into the creatures of California. Wow. One of the best parts of this event series is hearing the excitement conveyed by our researchers. I love a good science talk. And it's the thing that I most want to hear while I'm deaning. It sparks my intellectual curiosity, and it sparks hope to think about the fantastic minds thinking about and working on science questions. That undeniably includes our student researchers. The ecosystem of faculty, graduate students, and undergraduates is an interesting, complex, and highly interdependent one. It's not an exaggeration to say that our recruitment of the best faculty hinges on the quality of our graduate students. Faculty want to do work with the best students, which Berkeley has no shortage of. So here is my specific request of you, audience. The Integrative Biology Department has decided to present its students first as a place where you can direct your advocacy and support. Please consider making a gift as support in support of our graduate students, the glue of the research enterprise. Integrative Biology has kicked off a postcards from the field campaign to support its graduate students. I'm happy to say that I'm not only asking you without giving, and listen to this very carefully. In spring 2022, we plan to host a number of field trips with select faculty and student research groups where alumni and friends can come out with us on these field trips. Some of those faculty who have spoken here tonight will be hosts, and you've seen a small preview, and you will have names and faces for some of your hosts. Please watch for a future invitation and become a participant in a postcard from the field. I want to thank you for joining us today and encourage you to reach out if you would like to learn more about anything we've covered. We'll be sharing additional resources with you by email about today's topic, and speakers and a video of today's session will be available at basicscienceberkeley.edu a week from now. We look forward to seeing many of you at our next Basic Science Lights the Way event series and IB events, which will be in spring 2022. Until then, stay curious, fiat lux, and of course, go Bears!